Ever find yourself glued to those true crime documentaries, totally captivated by how DNA cracks the case?
Oh, absolutely.
Well, get this, just a few decades ago, the idea of using DNA to solve crimes seemed like something straight out of science fiction.
It really was groundbreaking.
Today, we're diving into how this revolutionary technology went from a scientist's hunch to a courtroom game changer, all thanks to these lecture notes you sent over.
And what a journey it's been. Let's just say the history of DNA forensics is full of plot twists you wouldn't believe.
Okay, so before we jump into the world of DNA, set the scene for us. What were investigators working with before this genetic revolution hit the scene?
Picture this. It's 1985, you're an investigator staring down a complex case.
Okay, I'm picturing it.
And your most reliable tools are often, well, eyewitness accounts and, if you're lucky, a bit of blood typing.
Eyewitness accounts. You mean like those blurry memories that could be swayed by like a strong gust of wind?
Exactly. And don't even get me started on blood typing.
And blood typing. I remember from these notes that in Jamaica, over half the population has type O blood. Talk about a needle in a haystack situation.
Precisely. It was like trying to solve a puzzle with only half the pieces, and they were all different shades of beige.
Right, not exactly CSI.
But then along came Sir Alec Jeffreys, a geneticist who famously said, I think people forget that it's not just about proving guilt, but about proving innocence.
And proving innocence was exactly what this new technology did in one of its very first applications. You mention a case from 1986, the murders of Dawn Ashworth and Linda Mann, where a man named Richard Buckland became the first person ever exonerated by DNA evidence. Can you tell us more about that?
Absolutely. Richard Buckland was the prime suspect. He knew one of the victims and had even confessed, falsely it turned out, under duress. But here's where the plot thickens. Jeffries had just discovered these unique repeating patterns in our DNA called VNTRs, variable number tandem repeats. These VNTRs, he realized, could be used to create a DNA fingerprint unique to each individual.
Hold on, back up a sec. DNA fingerprint. So it's like everyone's carrying around this invisible ID card written in their very cells.
Exactly. And when they compared Buckland's DNA fingerprint to the DNA found at the crime scene, law to match, he was cleared, the first of many exonerations thanks to this groundbreaking technology, but the real perpetrator was still out there.
Talk about a plot twist. So not only did DNA technology rewrite the rules of forensic science, but it also had this incredible power to right wrongful convictions right from the start. But how did they ultimately find the real killer in this case?
Well, they did something pretty bold. For the time, they launched a mass DNA screening of men in the area.
Wow.
Imagine the collecting DNA from thousands of individuals, hoping for a match.
Talk about a needle in a haystack.
It took a while, but eventually they found their guy, Colin Pitchfork. Faced with this irrefutable scientific evidence, he confessed.
Wow, that's amazing. But hold on, before DNA fingerprinting, before this case that basically launched 1,000 crime dramas, how did they even prove a family relationship? I remember this other case you from Christina Sarba and her son Andrew.
Yes. Christina and Andrew's case reminds us that DNA technology's impact goes far beyond just catching criminals. See, Andrew was detained by immigration, and they needed proof that Christina was his mother. This was back in the early days of DNA analysis, and the tests weren't as sophisticated as they are now.
Right, because the notes mention that while the test couldn't definitively confirm she was his mother, it did prove a familial relationship. And back then, that was a huge deal.
Exactly. It was groundbreaking and paved the way for using DNA evidence in immigration cases. And it highlights something crucial about why this technology is so powerful. DNA is everywhere. Literally, almost every cell in our body contains a copy of our unique genetic code. Blood, saliva, hair follicles, even bones. And it remains virtually unchanged throughout our lives.
It's like this hidden blueprint that tells the story of you, and we're just figuring out how to read it. But reading that story hasn't always been easy, right? The notes you shared mentioned something called RFLP analysis, which needed a lot of DNA and didn't work well with the graded samples. So how did scientists overcome those early limitations?
That's where PCR polymerase chain reaction bursts onto the scene.
Okay.
Now, this was a real game changer. Imagine being able to take a tiny snippet of DNA, like, say, a few cells left on a cigarette butt, and make millions of copies.
It's like finding a rare stamp in your attic and then suddenly having the power to make 1,000 replicas. You mentioned that PCR revolutionized everything. So how did this new abundance of DNA change the game?
Well, remember those VNTRs we talked about earlier? PCR made it possible to analyze even tiny amounts of VNTRs, opening up a whole new world of forensic possibilities. But scientists soon realized that while VNTRs were great, There was an even better option lurking in our DNA STRs, or short tandem repeats.
STRs, those are like VNTRs, but shorter, right? What's the big deal about these shorter repeats?
Think of it this way. Imagine you're trying to identify someone based on their fashion choices.
Okay.
VNTRs are like their entire outfit, while STRs are like the unique pattern on their scarf. STRs are shorter, more abundant, and they vary greatly from person to person, except, of course, for identical twins.
So focusing on those specific patterns, those STRs, gave scientists a more precise and reliable way to differentiate between individuals.
Exactly. STRs became the gold standard in DNA profiling, especially for something called CODIS. Think of CODIS like a massive, searchable database filled with the DNA fingerprints of convicted offenders and DNA collected from crime scenes. It's a powerful tool for linking crimes, identifying suspects, and even clearing innocent people.
It's incredible how one technology can have such a ripple effect, not only in solving crimes, but also in ensuring justice.
Absolutely. And remember, back in 2017, they made CODIS even more powerful by adding 7 new STR markers to the analysis. More markers mean greater accuracy, making it even harder for two unrelated individuals to have matching profiles.
Okay, so we've got PCR making millions of DNA copies, STRs giving us those unique genetic patterns, and CODIS acting like a giant searchable fingerprint database. But what about those cases where the DNA is super degraded, like in really old cases or with limited biological material? Is there a way around that?
That's where mitochondrial DNA, or mtDNA, comes in.
Okay.
Remember how I said almost every cell in our body contains DNA? Well, there's actually DNA outside the nucleus of the cell, too, inside these little powerhouses called mitochondria. And this mtDNA, it's inherited a bit differently.
Differently. How so?
While you inherit nuclear DNA from both parents, mtDNA comes solely from your mother. It's passed down through the maternal line like a secret code whispered from generation to generation.
So it's like tracing your ancestry back through your mother's line. But how does that help in forensic investigations?
Because it's present in hundreds or even thousands of copies per cell, there's a higher chance of finding usable mtDNA even in those severely degraded samples where traditional DNA analysis might fall short.
It's incredible how scientists have developed all these different tools to extract information from something as tiny as a strand of DNA. But with all these techniques available, RFLP, PCR, STR analysis, mtDNA, how do they decide which one to use?
That's a great question. It's like a detective's toolkit. With each technique offering its own strengths and weaknesses, the choice depends on a few factors, starting with the condition of the sample.
Right, like if it's old and degraded, mtDNA might be their best bet. But let's say we're dealing with a fresh sample from a crime scene. What happens once the investigators have carefully bagged and tagged that potential DNA evidence?
It all starts with extraction, basically, isolating the DNA from whatever they found, like a blood stain or a hair follicle. This step is crucial, and contamination is a big concern.
Right, like those crime shows where they're always yelling, "Don't contaminate the crime scene." Why is it so important to keep things squeaky clean when dealing with DNA?
We're talking about microscopic traces of genetic material here. Even a stray sneeze could introduce foreign DNA and throw off the entire investigation. Once they've carefully extracted the DNA, they need to figure out how much they've got to get. That's where DNA quantitation comes in.
Quantitation, so it's like measuring out the ingredients before you bake a cake, making sure you have just the right amount.
Precisely. Remember PCR, that magic trick where we make millions of DNA copies? Well, it only works within a specific concentration range. Too much DNA, and you get a messy, uninterpretable result. Too little, and the test might fail. Quantitation ensures they've got that Goldilocks amount, not too much, not too little, just right for PCR.
Precision is key. So they've extracted the DNA, measured it out carefully, and now it's time to hit the PCR button and make those millions of copies.
Exactly. And I'll often use those commercially available STR kits we talked about earlier. These kits have standardized the process, ensuring consistency and accuracy across different labs. It's like having a prepackaged recipe for DNA analysis.
So those kits are like prepackaged forensic magic. But once they've amplified those STRs, how do they actually visualize and interpret them? I mean, are we talking microscopic magnifying glasses here?
Not quite. They use a technique called electrophoresis, which separates the DNA fragments based on their size.
Okay.
It's like a microscopic race where smaller fragments zip through faster than larger ones. They use a special gel, and when they apply an electric current, the DNA fragments migrate through the gel, creating a unique pattern of bands.
Okay, so we've got separated DNA fragments, but how does that translate into a meaningful DNA profile? What do those results actually look like?
Think of it as a series of peaks on a graph. Each peak represents a specific STR allele, that's one of the possible forms the gene can take, and the position of the peak tells you its size. By comparing these peak patterns from different samples, they can determine if there's a match.
So it's like comparing those unique barcodes again. If the peaks align perfectly, it's a match. But what happens when they don't align? Does that automatically mean the samples are from different people?
Not necessarily.
Hmm.
There are a few possibilities. It could be an exclusion, meaning the samples are definitely from different sources, which is helpful information in itself. Or it could be an inconclusive result where the data isn't clear enough to make a definitive call.
So it's not always a clear-cut aha moment sometimes. The DNA evidence is more like a cryptic clue that requires further investigation.
Absolutely. And even when it does lead to a match, there's another layer of analysis involved. Statistical analysis. They need to determine the probability of someone else having the same DNA profile. The more STRs they analyze, the lower that probability becomes.
So it's about reducing those odds to practically zero, making that match incredibly strong evidence. And we've come a long way from those early days of DNA fingerprinting to today's high-tech analysis. But as technology continues to evolve at lightning speed, what does the future hold for the world of DNA forensics?
Well, there's one advancement in particular that has the potential to revolutionize the field once again, next generation sequencing, or NGS. And let me tell you, it's straight out of a sci-fi movie.
NGS. Okay, you're going to have to break that one down first. What is next generation sequencing and how might it impact the world of forensics? NGS even sounds futuristic. So what is it exactly? Is it like those scanners you see in sci-fi movies that analyze someone's entire genetic makeup in seconds?
You're not far off. NGS takes those techniques we've been talking about, PCR, STR analysis, and kicks them into overdrive. It allows scientists to sequence massive amounts of DNA faster, cheaper, and in much greater detail than ever before.
Okay, so we're talking about a whole new level of detail here. But what does that mean for solving crimes? Instead of just those specific STR markers, could they analyze, like, everything?
Exactly. We're talking about potentially identifying everything from someone's hair and eye color to their ancestry and even predispositions to certain diseases, all from a tiny DNA sample. It's like upgrading from those old pixelated security camera images to ultra-high definition.
Wow, that's both incredible and a little bit unnerving, right? I mean, on the one hand, you've got this incredible tool that could solve cold cases and crack mysteries we thought were unsolvable. But on the other hand, it feels like a huge responsibility. Where do we draw the line with this kind of information?
That's the million-dollar question, isn't it?
Yeah.
NGS has the potential to revolutionize forensics, but it also presents us with some profound ethical and legal challenges. How do we ensure this technology is used responsibly, protecting individual privacy, while also maximizing its potential to bring justice?
It's like opening a door to a whole new world, and we need to make sure we're prepared for what we might find on the other side. But for all the unknowns, it seems like this DNA journey from those early days of blood typing to the brink of this NGS revolution has fundamentally changed the way we understand crime, justice, and even ourselves.
Absolutely. And it really underscores the power of scientific curiosity, the importance of constantly questioning and pushing boundaries. Who knows what incredible advancements the future holds? But one thing's for sure, as we unlock more secrets hidden within our DNA, we'll need to proceed with both enthusiasm and caution, ensuring these powerful tools are always used for good.
It's been quite a deep dive into the world of DNA forensics. We've gone from blurry fingerprints to genetic fingerprints, from relying on eyewitness accounts to decoding the very building blocks of life. It's incredible to think that all this information was hidden in plain sight, just waiting for science to catch up.
And as we wrap up, I have a question for you to ponder. As this technology continues to advance, what new ethical dilemmas might arise? And how do you think we, as a society, should navigate those challenges?
It's a question worth considering, and one that I'll definitely be thinking about long after this deep dive. Until next time, keep exploring, and remember, sometimes the most profound discoveries are hidden in the most unexpected places.